Welcome students to Micromed Education. I'm Dr. Rachna Solanki and today we are going to discuss regarding the bacterial morphology. However, the chapter is very vast, so it's difficult to complete within the one session. So I have breakdown into that, and today we are going to discuss regarding Fimbria and Flagella. So what do you mean by Fimbria and Flagella? So the Fimbria is an organ of adhesion. It is also called as pili, and some hair-like protein appendages that is present on the bacterial cell that will allow or to adhere to the epithelial cells. This is called common pili. Then another is called sex pili. It is basically a longer, as shown in this image, that facilitate conjugation and transferring the plasmid DNA between the bacteria. This is another way how the bacteria acquire antibiotic resistance. So if we consider what is the clinical signification for the Fimbria, so basically like E. coli, it is used to attachment of the bladder epithelial cells causing urinary tract infection, and Neisseria gonorrhoeae that will attach to the urogenital mucosa and leads to gonorrhea. So remember, Fimbria is a pili or hair-like appendages that will allow bacteria to attach to the epithelium. Now flagella. Flagella is a whip-like structure and it is an organ of locomotion. So it has a three part: filaments, then hook, and the basal wall. Filaments is a long helical protein extending from the cell. Hook, it is a flexible compound connecting filaments to the basal body, and basal body it's basically embedded into the cell envelope powered by a motive force, proton motive force. What are the clinical significance or importance of flagella? First, as it is an organ of locomotion, it allows bacteria to, in the movement, provides motility. It helps in the spread and invasion of within the tissue, as well as it is useful for the serological diagnosis like at H antigen in Salmonella group. Based on the arrangement of the flagella, they are divided into monotrichous, lophotrichous, amphitrichous, peritrichous, and atrichous. So mono means only one at one pole. An example is Vibrio cholerae. Lopho is a tuft and it is present in the spiralium. Amphi is at the both end, one side on this and this side. Either it can be a tuft or a single, that Alpigens faecalis. Peritrichous, it is all over, and atrichous, it is without. That is of Klebsiella pneumoniae. Now, based on the flagella and the organ, different type of motility is there. So there are two mainly questions asked: what is the importance of the flagella and the arrangement of the flagella? So it asks into 2 marks. Then even the types of motility. So tumbling motility, मतलब कुलाटी मारना जो बोलते हैं उसे tumbling motility है, that is by Listeria monocytogenes. Gliding motility by mycoplasma, stately by Clostridia. Darting मतलब वो भाला जो डालते हैं ना, it is called dart. But darting motility is by Vibrio cholerae and Campylobacter, which is a very fast movement. Swarming, पानी में पत्थर डालने पे जो concentric rings forms होती है, वैसी टाइप की motility on blood agar plate, it's by protease and Clostridium tetani. And corkscrew or flexion extension type of motility is of spirochetes. So these are the important things that is mainly asked from the flagella. And then let's revise for the fast that Fimbria is hair-like short appendage, helpful in the attachment, colonization as well as in conjugation process. Flagella is an organ of locomotion, whip-like appendage, monotrichous singly, lopho at the tuft spiralium, amphitrichous at one end or both, one at each end, that is Alpigens faecalis, peritrichous, that is all over by E. coli, and atrichous without flagella, that is Klebsiella pneumoniae. Hope this find useful to you, and we'll see you in the next lecture. The link of the same where you can download the PDF is inside in the description. Stay tuned more lectures to the microbiome education. Bye.